So our second speaker today is Nithya Ramakrishnan. So Nithya did her PhD at the Indian Institute of Technology in Delhi with Ranjan Bose, studying advanced information theory for analysis of biological data. And she went on from there to do postdocs at the Indian Institute of Technology, Bombay, as well as Dalhousie University in Canada, and a senior project scientist role with Indian Institute of Technology, Guwahati. Before she started as an assistant professor at the Indian Institute of Bioinformatics and Applied Biotechnology in Bengaluru in 2023, where she applies machine learning, deep learning, information theoretic, and algorithmic techniques to model biological problems and analyze biological data sets. And I'm very excited to hear her talk today on how DNA replication is a noisy communication channel. So please join me in giving a warm, albeit virtual, welcome to our second speaker today. Nithya, the floor is yours.
Yeah, thank you, Ben, and thank you for this opportunity. So good evening to all joining from India, though late, and good morning to everyone. Today my talk is going to be on DNA replication being a noisy communication channel. So let's look at what are the motivations. Primarily, we intend to model the process of inheritance of the epigenetic modifications as a noisy communication channel. And the idea is to see how well the daughter chromatids inherit the patterns of the mother chromatid. Now, I would like to start with a brief primer on the basics for those who are not from the biology background. Since I am basically a computational person, I would just like to set the floor even. So we know the genetic sequence information is there in DNA. It is common across all the cell types, whereas it is the epigenetic information that could encompass DNA methylation, non-coding RNAs, or histone post-translational modifications, which actually primarily decide the switching on or off of the genes and the differentiation of cells as well. So today we'll be focusing on histone PTMs and how they are inherited from the mother to the daughter. So again, we know that nucleosome is an octamer, and it was originally considered to be the packaging units in the cell with 147 base pairs of DNA wrapped around it. So we have 4 types of histones: H3, H4, H2A, and H2B. So when we consider a modification, what I mean by a histone PTM is a particular amino acid on the tail of, let's say, H3 being trimethylated is H3K27 Me3. And we know that this is one of the most important repressive modifications, which is responsible for turning off the gene. And also from literature, it is well established that these histone PTMs, there are many. It could encompass methylations or acetylations or ubiquitinations in specific amino acids in different tails of the 4 kinds of histones. They follow patterns across the chromatid. So for instance, activating modifications or promoter modifications like H3K56 are enhanced near the TSS, whereas those like K36Me3 might be enhanced in the gene bodies. Now, it is quite critical that these patterns are preserved from the mother to the daughter chromatids when the DNA is actually replicating. So how does the enzyme machinery ensure, and the histone chaperones, all those biological complexes, how do they actually ensure that this pattern is inherited with fidelity? So that is a question we set out to ask and answer. So first, we start off by modeling the mother chromatin as a string of beads. So each nucleosome is considered to be a bit with either 1 or 0, representing the presence or absence of a particular modification in question. So we start off with only considering one modification, say H3K27 trimethyl. So if the modification is present in the nucleosome, then it's a 1 or it's a 0. So this simple walk across the chromatin is modeled as a binary valued random walk, and we represent it as a Markov, first order Markov model with probabilities alpha and beta representing the probability of a 1 staying in a 1 or a 0 staying in a 0. That is, if you are currently in one nucleosome, which is having the modification, and you jump to the next one, which also has a modification, then it is with probability alpha. Likewise, it's probability beta for a 0 to 0, and the transition probabilities are 1 minus alpha and 1 minus beta. So largely speaking, what alpha beta here represents in the biological context is the run lengths or the ones of the modification presence or absence. So which means if values of alpha and beta are very high, you have long runs of ones and long runs of zeros. So after this, we just take a small primer on how the communication channel works. So as you know, a source signal from, let's say, a phone is actually, how does it reach your friend's phone, which is your destination? It is through the wireless channel, which corrupts the signal, as you can see. So this corrupted signal is decoded at the receiving end using a variety of algorithms. So knowing that this is probably the kind of noise that might be there, and this is probably the kind of signal that could have been there. So the actual signal is decoded by the destination decoder. So this principle of communication theory is now applied to the DNA replication process based on certain observations from experiments. So a set of experiments conducted by different labs tell us that when ahead of the replication fork, the parental nucleosomes are dislodged and they set out as H3H4 tetramers, and they are placed randomly in one of the daughter chromatids, and that is with equal bias, which means either the leading or the lagging strands get this H3H4 tetramers with equal probability. So this is considering canonical histones, not variant histones, and we're also considering a cell without any mutations. For instance, those MCM2 might be the chaperones responsible for ensuring that there's no bias between the leading and the lagging strands, and it is symmetric inheritance. So we are considering these assumptions and going forward with this simplest model in the beginning. So when the parental tetramers are placed on either of the daughter strands, but at random positions close to the replication fork, because we know again from literature that repressive modifications like K27 trimethyl are placed very close to the replication fork, but at random positions in the chromatid. So each daughter chromatid just inherits 50% of the parental nucleosomes. But still, it has to inherit the exact pattern of the mother chromatid before the replication. So how does that happen? So with only 50% of the information, how does the rest of the information recover? So this is the actual question. And we also know this, that from a histone pool, newly formed tetramers are placed in those positions where the parents are not there. But those newly formed tetramers do not have the modifications. So it's actually as if you have just a few ones and a lot of zeros. So the zeros are going to be modified based on the neighboring ones. But whether to modify them or not, that is the question. Because is that particular nucleosome originally modified in the mother or not? So to answer this question, just think of this mother sequence having a set of ones. And ones, of course, represent the modification presence. And each of these blue ones are nucleosomes are gone to either of the daughter chromatids with equal probability. And the pink zeros are the ones that come from the new histone pools, and they're all zeros because they don't have the modification to start off. Now the enzyme machinery has to modify these zeros based on the neighboring patterns. So whether if the neighbor has it, should it always be a 1 or not? So these questions, how do they get answered? So we propose that the enzyme machinery follows a set of algorithms and tries to reconstruct the mother chromatin from the daughter chromatin. So here's the model. So the DNA replication is represented as this, as you can see, a noisy communication channel. where the mother sequence is ANDed with Z here. Z represents a noisy signal. It is just a set of zeros which gets ANDed to the mother, and it converts a few ones to zeros, because that's why the daughter has lost some of his ones. And this daughter is now decoded to restore M, which we call as M cap, which has to be as close as possible to the mother. So the cartoon there also represents how the daughter sequence D has got a lot of red zeros. However, upon correction, you find that some of the zeros are restored back to ones, right? So again, the channel diagram on the top right represents, we call it Z channel, where the mother could have both one and zeros, but it is actually converted. The one could be flipped to a zero with probability 50%. So now to recover a set of zeros between two ones, we employ a probabilistic technique. So the idea is to recover the signal based on which of the combinations would give you the maximum probability in the recovery. So if a pattern is, if it's an all one pattern, which is giving you the highest probability of recovery, then it is chosen. Or if it is a all zero pattern between two ones, or if it is a 1-0-1-0 pattern. So which of these patterns do give the highest probability? That's so to do that, we have a Viterbi algorithm here, and that is based on the maximum a posteriori probability, or the MAP algorithm, MAP concept. So using that, we try to recover the mother signal. And the branch probabilities, as you can see, are based on the statistical parameters of the mother chromatin. They could be alpha, beta, which represent the probabilities of one being in a one or a zero being in a zero. So using that, we see the fidelity of the inheritance of the reconstructed mother from the original mother. Now, to quantify it, we have what is called the BER, or the bit error rate. So what is a BER denoting here? It denotes the error between the original mother and the reconstructed mother after Viterbi decoding. Now, as you can see, this BER is dependent on alpha beta, because the Viterbi algorithm works based on alpha beta. So for different regions of alpha beta, we find that the BER varies with from 5 to 30%. So when and beta are either too high or too low, the BER is very less, but if it is in the medium range, it's around 30%. So we also try to decode it into this phase diagram that you see on the right. And we see that different regions, like in A, any zero between the ones are always filled. So when alpha is very high compared to beta, any new histones that are between the parental nucleosomes are always modified. Whereas in region B, when both alpha and beta are very high, we find that when there is a stretch of nucleosomes, new nucleosomes between bordering parental nucleosomes, they are modified only if the run length is less than a particular threshold. That is, the threshold is given by the values of alpha and beta using this formula. So to make this more clear, consider a daughter chromatin that is newly formed, and consider that it has now some parental nucleosomes which have some modifications which are randomly placed in it. Now, some newly formed nucleosomes are placed in between them, and the enzyme machinery now has to decide which ones to modify. Now, it will modify only those ones that are within a particular range. The gap length must be within a particular range. Now, in this case, the first two cases, the gap length was only 2 and 1, so they got modified. Whereas here, the gap length is 6. So if it decides not to fill this, because probably according to this alpha beta, the K was less than 5 or less than 5. So it is decided not to do this. So this is just a cartoon which illustrates what we call as threshold K filling. So we validated this model from experimental data obtained for H3K27Me3 from this site, from the Groth lab. So we binarized the chromatin in this range, as mentioned here, and we made it undergo the loss to produce a daughter chromatin, and we reconstructed it with different K values of K. So according to the alpha beta in this range, the value of K that produced the lowest error was 5, as you can see from the violin plot below. And indeed, it was with this 5 value of K that we got the best fidelity. So this actually proved that this model works as expected in the particularly in the high regions of alpha and beta, as shown in the phase plot. Now, with this, we proceed on to the next question that we want to ask. So far, we were dealing with only one modification of interest, but as we know, there are several modifications which work in tandem. Now we go on to one more significant modification, which is primarily antagonistic to the primary modification. So for instance, K27ME3, we know is antagonistic to K36ME3, which is an activating modification. Now, what if some nucleosomes which are neighboring to K27ME3 had this antagonistic modification? Will the inheritance actually be benefited? Will the inheritance of K27ME3 benefit from the presence of K36ME3 in the neighbors? If yes, by how much? So that's a question we set out to answer next. So now, as you can see, there are two binary valued random walks, one representing the modification A, MA, and the other representing modification B. And consequently, we have the Z channel now becoming what we call as a binary erasure channel. So which means the daughter chromatin can have either a 1, which represents a modification PTM-A, or it can have a 0, which means there is no modification, or it can have a 2, which means it does not have modification A, but it has modification B. Now, the presence of 2 is adding more information to the enzyme machinery. So ideally, we should expect the presence of antagonism to improve the fidelity of K27ME3. So does that actually happen? So we again use the Markov model, which was initially a 2-state thing, has now become a 3-state Markov model. We have an additional parameter mu, which represents antagonism. That is, we consider mu to be the probability of modification B being present when modification A is absent. So we also assume that no nucleosome is bivalent, as a no nucleosome is carrying both the modifications together. So it has either one of them or none at all. So with these assumptions, we go and perform the Viterbi decoding. But this time, as you can see, there are 4 different trellis diagrams, because in the earlier case, we just considered only one to be the borders. Here we have one or the second modification as well, one representing PTM-A, whereas the other representing PTM-B. They could be the borders, or both of them could be PTM-Bs, or one on the either side. So, so these 4 trellis diagrams could be the 4 cases of decoding. And here the value of K, threshold K, was indeed present in the same region, but then it was modified. Mathematically, we got the value to be inclusive of mu as expected. And we drew out the heat maps, and we also got the Hinton plots, comparing the case of no antagonism versus the case of antagonism for different values. So when mu is equal to 0.2, it means that only 20% of the nucleosomes that do not have modification A have modification B. And whereas if mu is equal to 0.8, then 80% of those that do not have modification A have modification B. So if this antagonism increases, then the fidelity was improving. So that is why the size of the largest square, as you can see, keeps on increasing, because the fidelity in the Hinton plots, because the BR was decreasing with increasing antagonism. Okay, so this was also proved using this violin plot obtained from the mouse embryonic stem cell data, with K27ME3 being the primary modification of interest, which is PTM-A, and K36ME3 being the PTM-B, which is the antagonistic modification. Now, what does this actually mean? From the biological context, what we are proposing is that regions which have antagonistic modifications in their neighbors, which is poised chromatin, are inherited with much higher fidelity than those without it. Now, the next approach is an alternate way instead of Viterbi decoding. Can this be decoded using some other techniques, primarily a machine learning algorithm such as neural networks? Now, why would one go for such a thing? Primarily, when we consider Viterbi decoding, it is from when we went on from one modification to two, the complexity increased. So when we go in for multiple modifications at the same time, how does the enzyme machinery decide whether to modify a particular de novo nucleosome or not? So it could be a complex set of factors which we thought could be learned using a neural network, which might model the memory aspect of the mother chromatin. So we again consider two cases, one without antagonism, which you see on the left, and one with antagonism that you see on the right. So in both the cases, we simulated 100 nucleosomes, the daughter chromatin with the loss of 50% of its mother as the input, and we try to decode the retrieved mother chromatin as the output. So this is after the hidden layers, which were like, we had about 5 hidden layers, each of them with 150 nodes. So with the presence of antagonistic modifications, where we had 200 input nucleosomes, when predicted the mother MA from DAB, we, as expected, found better BER. BER was the same as before, as compared to the case without the antagonistic modifications. So these are some of the neural network parameters. So this is again an illustrative, this is again the heat map results from the neural networks, which also illustrates that how the presence of the antagonism greatly improves the fidelity, as shown by the black squares, which indicate the difference between the antagonistic and non-antagonistic cases. So these neural networks also were employed to predict alpha, beta, and mu, the model parameters given any mother sequence, and the combination of mother sequence A as well as mother sequence B. So there we showed that there's a high correlation between alpha and the predicted and the actual values of alpha, beta, and mu. So any given chromatin, if could be statistically analyzed, or the parameters could be learned using a machine learning model, is what we also showed. Now, like I mentioned before, why neural networks? When we have more deterministic algorithms like Viterbi, in case you want to model the replication process as a noisy communication channel, when we want to analyze the effect of multiple histone PTMs, and also to combine the effects of other epigenetic markers such as DNA methylations and non-coding RNAs and other transcription factors, which might all work in tandem to help in the inheritance across a cell cycle. Because we are studying the replication process immediately after the cell cycle, which we call as primary restoration. This is definitely followed by several secondary restoration mechanisms. To model most of them, we could employ further machine learning algorithms, as suggested. So as part of the future work, we plan to include other variations to the model, like for instance, histone variants such as 3.3, which have certain affinity to certain types of modifications, and also which do not split as tetramers but as dimers and undergo intranucleosomal copying, and also to model the inheritance over multiple cell cycles, leading to a certain loss in the epigenetic information from across generations. Does that result in aging? And of course, the impact on the diseases. So I want to thank my mentors, Professor Sibiraj Pillai and Professor Ranjit from IIT Bombay. And the work from the antagonistic part and the neural networks was done by Balu. He's currently at University of Bonn. And I'm always grateful to my lab members, colleagues, and friends at IBAM. Thank you, and I can take questions.
Thank you so much, Nithya. That was fantastic. So for people in the audience, you can type your questions into the Q&A box or raise your hand and I'll unmute you to ask your question yourself. But while that's happening, I have a few questions to start with. So I I was wondering, the values that you're seeing for kind of the noisy replication or inheritance, how much do you think those will change across cell types or across kind of environmental conditions?
Yeah, that's a great question. So we are considering normal mitotic cell division, where, like I mentioned, there are certain parameters like symmetric inheritance, and we do not consider histone variants at this point. But those might not be true for example, say meiotic cell division,
Mm,
when stem cells are actually asymmetrically dividing. So the model itself might not be applicable in those cases. So those are things that we are working on, but this we believe is, let's say, largely true for most of the cells which are undergoing mitosis. So
yeah, no, that's cool. And then, I mean, I guess what I was wondering about is like, would you expect changes in something like a cancer cell to alter the fidelity of this process? Like, could it get noisier or less noisy?
So cancer cells, actually, the fidelity is quite high because
Mm,
they do copy and very fast from the parents. And so whereas if you consider certain things like stem cells, it might be very different, especially since it is going to be asymmetrically dividing.
mm,
So there is a lot of difference across different cell types for certain. Yeah, we are studying them as specialized cases of this generalized model, like I mentioned, as part of the future work.
excellent. And then, um, ca- are you thinking about how to work into the model? Like for some of these modifications, there are mechanisms to kind of reestablish them quite quickly. So kind of like how much of what you're seeing is inheritance versus reestablishment of the patterns de novo.
Yeah, so if you mean something like, for instance, K27ME3 is one of the repressive modifications which we know is inherited immediately upon replication.
Hmm.
So, as opposed to certain activating modifications, which might actually be re-established over this period of the cell cycle, like I did mention the variant histones H3 dot to be coming and
Hmm.
taking over different time points over the cell cycle. So, and which also has an affinity to such activating modifications. So that's why most of our experimental validations is also based on K27 trimethyl at this point of
Hmm.
time. So when we consider variant histones and the secondary mechanisms, we will be looking at that re-establishment, which you mentioned, which involves K4ME3 or other activating modifications.
Excellent. And then, so, in the work you presented, you focused on like H3 and H4 and the tetramer. Um, have you thought much, or are you thinking of looking at H2B and how that is inherited or not through the cell cycle?
It is an ongoing work, but see, it's since we are primarily working with biologists and all that,
Hmm.
but it's first currently we want to exhaust the most important ones like involved with H3 and H4, and because I believe from what I understand the mechanisms of inheritance of the dimers in H2A H2B are entirely different, so
Hmm.
it would intranucleosomal copying as opposed to inter. So in which case the whole thing has to be altered accordingly. So yeah, we'll get there when we get there. So
Yeah,
that's
yeah, yeah. No, that makes sense. And then a last question for me is, have you looked at any like it feels like every year there's another paper describing more kind of oncahistone mutations.
yeah.
And so have you thought about looking at whether some of these oncahistone mutations affect the inheritance of the histones themselves?
Yeah, in fact, we were looking at K27M mutations
Mm,
in the IPG because that particularly affected the antagonism between K27ME3 and K36ME3 to a large extent, which the antagonism was lost. So this was actually directly applicable as a sort of an extension to our model. But again, yeah, you kind of read my mind. So it's yeah, it's in the works, yeah, in the
Yeah,
pipeline.
excellent. Well, thank
Okay.
you very much for the wonderful seminar, and thank you to Julia as well, and thank you everyone for attending. I hope you all have a happy holidays. We will be back towards the end of January, so keep your eyes peeled for the registration links and the sign up for speakers in the new year. But thank you again to both our speakers today, and thank you everyone for joining.
Thank you.